Right. All right. Hi, everyone. I'd like to introduce our special speaker for today, Dr. Jessica Hite. Dr. Hite is currently an assistant professor at the School of Veterinary Medicine at the University of Wisconsin-Madison. We first met when we were postdocs, and even back then, she was clearly already one of the leading researchers working at the interface of ecology, evolutionary biology, and disease management. And to be honest, I had kind of a tough time trying to boil all her research profile down to sentences, given that it is really interdisciplinary. As an NIH postdoc fellow, wow, Dr. Hite worked with conservation groups in the Smithsonian Tropical Research Institute to identify environmental factors driving biodiversity and disease outbreaks in aquatic systems. She's also been highly active in agricultural research and has played a critical part in shaping our understanding of bioenergetics and drug resistance. Illness-induced anorexia is just one of the many landmark ideas she has helped to advance. Right now, Dr. Hite is extending her work to human systems, looking at how various sociological factors, including nutritional availability, can impact epidemics in parts of the world where resources are. So we're really lucky to have such a pioneer researcher with us today. And for sports fans, to feel like catching Caitlin Clark play in person. And now please give her a very
Thank you, Yoon, for that very generous introduction. I promise no Clarkian impressiveness here today. Thank you all very much for joining me to learn about these evolutionary puzzles and ecological kaleidoscopes that I have been pondering over the last few years. And before we get started, I'd like to acknowledge that this work is part of a great collaborative team, including Dr. Clay Kressler, Dr. Laurel Lagenza, folks from my lab, including two postdocs, Chloe Fuyu and Colette Nicodem, and a really outstanding lab scientist, Eric, Eckwall, who is not a fan of photos, so gets to be my favorite bird instead. And the puzzle that we will ponder today is, why isn't drug resistance more of a problem? So if you're in this department, I'm sure that you are all aware of drug resistance being a major public health challenge, right? Before COVID, it was named by the World Health Organization as the #1 public health threat, right? So it's a problem. And most of our experiments and theory designed to understand the emergence and persistence of antibiotic resistance and other traits like virulence that undermine our most powerful biomedical tools assume that these traits carry large fitness costs. Now, these costs are necessary, but not sufficient to explain patterns of either virulence or drug resistance. So if we take the case of drug resistance, for example, it's everywhere. It's right up there with microplastics as the #1 environmental pollutant that is insidious and goes undetected because we can't see it with our naked eyes, right? So we have antibiotic resistance genes in polar bears in Norway, in mountain lakes in the Tibetan Plateau, and in wild fish populations on Vancouver Island. In my backyard in Wisconsin, 52% of the cows that we sampled had multi-drug resistant E. coli. And because what happens on the farm definitely does not stay on them. When we looked across canine and human samples, we found similar patterns, although at lower rates. And then when we investigate the genetic signatures of these E. coli, we find that there's overlap between these different hosts. And that's because through the wonders of horizontal gene transfer, antibiotic resistance genes, plasmids, can move across boundaries. So they can move from E. coli to E. coli, E. coli to salmonella, salmonella in a cow, over to salmonella in a human, and over to your dog. And we have increasing evidence of this cross-species transmission. What we don't quite understand is how long it's sticking around and if it's causing problems later on in terms of fitness, for wild populations, or in terms of public health threats. For example, do farmers that harbor these antimicrobial resistance genes in their gut microbiome have a higher likelihood of acquiring a drug-resistant infection later on? These are big open questions. And this leads us back to our original question of, if there's all this cross-species transmission, and if antimicrobial resistance genes are everywhere in the environment, why doesn't everyone have a drug-resistant infection? Why are drug-resistant infections severe in only certain parts of the world, or problematic in only certain patients, say for instance, those with other comorbidities? Until we can address this question, we're having a really hard time at moving on documentation. So we're really good at documenting where antimicrobial resistance genes pop up, how they move across species. We don't have a good understanding of the other environmental drivers that are behind those patterns, and that hinders our ability to accurately predict future emergences. So we'll explore that today using field data, mathematical models, and if I talk fast at the right pace, we will also look at some experiments that we're doing to test these mathematical predictions. And the key takeaway from all of that, just to prep you, is that variation matters. So variation within host interactions between the immune system and pathogens, as well as colonization efficacy, can generate variation in pathogen traits through space and time. And this variation and ecological interactions can either suppress or create niche space for these virulent or resistant pathogens. So in other words, the patterns of drug resistance, these are emergent properties of the complex microbial communities in which these pathogens are embedded. And if you're an ecologist, that seems obvious, right? If you want to think about species diversity or species coexistence in a prairie, it would make sense for you to study the entire community, not just an individual plant or an individual genotype. But that's basically how we've studied antimicrobial resistance in the lab. We've taken a single isolate, we placed it in an ideal condition all by itself, and then we've asked, how does drug resistance evolve under these ideal conditions? And the emerging view is that we need to take this more complex ecological perspective to really address this question. So we uncover these emergent properties by linking eco-evolutionary dynamics across multiple scales. And this is where research in my lab is focused over the last few years, and also in my postdoc. And we've done that in part by opening the within host black box, right? So because I'm an ecologist by training, everything comes down to consumer resource interactions and competition for resources. That explains all of the dynamics in natural systems that we see, right? So that's where I always start, and I always start with theory for those consumer resource interactions. And in this case, what we've done is we just use a series of coupled ordinary differential equations to basically look at the food web within hosts. And instead of there being predators and prey in this food web, we now have resources that the host uses for its own growth, maintenance needs, like reproduction and development. And then pathogens steal those resources for their own growth and development, which then causes hosts to have to allocate some of those maintenance costs over to costly immune defense. And these within host interactions scale up to influence transmission between hosts at the herd level. And this determines, say, shedding rates into the environment, the size of the epidemic, which then feeds back in to influence individual infection dynamics. Now, because of COVID, you're all acutely aware of these important connections, right? For some individuals having more severe infections, shedding more propagules into the environment, and that having these cascading effects across the herd level. So hopefully connecting across these different biological scales makes intuitive sense. And you're also aware that if we want to understand pathogen evolution, we're going to think about multiple traits of these pathogens, multiple types of viral strains, for example, in the case of COVID, right? We can do the same thing when we're talking about drug-resistant and virulent strains. So in this case, we would use an adaptive dynamics framework to plug it into this standard epidemiological model. And we ask, if we have a resident strain with a given set of traits, say a certain level of drug resistance or a certain level of virulence, and we introduce a mutant with a slightly different set of traits, say it's a little more resistant or a little more virulent, and we run this over multiple generations, can that mutant strain become established in the population to the point that it takes over and becomes the new resident? And in adaptive dynamics framework, you do that by identifying the evolutionarily stable strategy. Okay, so we've connected across these different scales. We've now connected time scales. So ecological and evolutionary processes are happening on the same time scale. And we've also connected within host and between host dynamics, right? And that's important because pathogens have to be successful across all of these different scales, which is pretty impressive, right? That they're able to quickly adapt to all of these different environments. And all of these connections highlight that there's tons of feedback loops, right? So all of these different feedback loops have important consequences for the dynamical aspects of the system. And importantly, they create tons of opportunities for variation in those within host dynamics, right? So individuals are going to differ in their infection outcomes based on their sex, their age, their prior exposure, their immune status, et cetera, et cetera. And in our models, a lot of those within host dynamics are mechanistically linked to their resource condition, right? So higher resources typically correlate with higher immune functioning, and there's also then more resources for the pathogens to steal. So this creates tension across those different competing interests. And what this highlights is that optimality, which is how we determine these evolutionarily stable strategies, for a pathogen trait within a host can differ at the between host level, right? Most approaches, so our standard SIR departmental models, do not account for the variation at these different levels because they abstract out the individual level dynamics. So these within host interactions, they just assume that they go to equilibrium rapidly. The ecological interactions, they go to equilibrium rapidly. And so all of that is abstracted out. What this implies is that within host dynamics and infection outcomes are on average identical across all hosts. And that's a simplification that we've made largely because we had to, with limited computational power, we don't have to make that simplification anymore, especially since we're aware that individual level variation is very real and it matters, right? We all know that on a deep level, given what we've all just been through with the pandemic, right? Individual level variation matters, and we'd like that to be accounted for in our models. And from sort of a theoretical perspective, these positive feedback loops have lots of implications for the system dynamics. In particular, they can lead to multi-stability. And multi-stability is really interesting when we're talking about the maintenance of genetic variation and species coexistence. So if we plug all of those cartoon interactions into a mathematical model, and we look at how the gradient of resources influence the evolution of a pathogen trait, in this case, we're looking at exploitation rate. And that's because we're assuming that traits like virulence and drug resistance are energetically costly to build and maintain. And so pathogens that need those extra resources are then going to exploit more resources from their host, right? So higher exploitation rate is going to facilitate the emergence of traits like drug resistance and virulence. And what we find is multiple steady states. So in this case, we have multiple convergent stable ESS. And we have a high virulence strain that's coexisting with a low virulence strain across a pretty wide gradient of resource levels. And this high virulence strain has stabilizing effect on these eco-evolutionary dynamics, whereas the low virulence strain does not stabilize. And in other words, the coexistence of these different traits feeds back to influence these eco-evolutionary dynamics that affects species coexistence among these different pathogen strains. And the other aspect of all of these multiple feedback loops is that it implies that the system is very sensitive to the initial starting conditions. So the initial resource condition of the host that impacts its immune dynamics or its initial exposure to pathogens, all of these initial conditions affect the ultimate evolutionary trajectory of the entire system. So what are other classic examples of bimodality in ecology that you might be familiar with? Well, strong intraspecific competition, for example, in simple Lagrange models, which leads to mutual inhibition, are known to drive these different modes of multistability. And so this also harkens back to the importance of thinking about competition among different microbes and mechanistically connecting these to resource interactions. So again, we have this kind of foundational ecology theory underlying the epidemiological and evolutionary dynamics, which just means that we have a lot more theory and experimental evidence to draw on to help us develop these models, to interpret the results, and then also to connect those to experiments and field data. Okay, so let's talk about other consequences of these multiple feedback loops and why it's important to open this within host black box, again, thinking about the coexistence of these different kinds of traits, which we've made the assumption again that they're energetically costly. So the importance of these connections on a different level has to do with whether or not we're interested at the between host or the within host level, right? So from a managerial perspective, if we're treating our herd to reduce disease, we have to make the decision of whether or not we're going to be treating for individuals or reducing outbreaks at the population level, right? And that matters for the evolution of these traits because what's optimal at the between host level is going to be the strain that infects more hosts than the other, whereas at the individual level, it's the strain that reaches the highest density. And the highest density in this case is going to translate to virulence, right? So I'm going to show you the consequences of this in a couple of different ways. All right, so the conclusions from part one is that these positive feedback loops, which we've looked at and are facilitated by linking multiple scales of biological organization, can lead to multi-stability. In this case, multi-stability helps us understand why we can have variation in traits that we would assume would just reach equilibrium and outcompete other traits, right? So if we didn't have multi-stability, what we would expect to see is just drug-resistant traits outcompeting every other strain, and all of our infections are now drug-resistant, or they're all highly virulent. So this multi-stability helps create genetic diversity that allows other competitive interactions to keep those more toxic traits in check. And understanding these patterns and this variation in pathogen dynamics might help us understand variation across broad spatial scales. So the question is, can this help us inform management decisions, right? So this is all interesting theoretically. We've already seen that we have drug resistance everywhere, and it varies really broadly. Great, we have some explanations for that. Can we use this information to help constrain drug resistance so that it doesn't become a continually increasing urgent public health? So if we go back to our data from drug-resistant E. coli in dairy, canines, and humans, we might ask that logically, if we want to limit drug resistance, we might start to work on the sector that uses the most antimicrobials and limit those with stewardship programs, right? So this is why we need a prescription in many parts of the world to acquire antimicrobials, right? It's important to note that most of the world does not require prescriptions, but here this has been our primary strategy is limit use of antimicrobials to protect their lifespan, right? Their shelf life, essentially. And this is important because we know that there's a strong correlation between drug use and the prevalence of resistance, right? So this pattern is very sensitive. If we increase drug use, we see patterns of resistance go up. But similarly, if we limit drug use, we see patterns of resistance go down. And some of the best data linking these patterns comes from Scandinavia, where in livestock systems, they've outlawed specific drugs. You can see those patterns decrease rapidly, like within the next year of that policy being implanted, right? So these patterns are really strong. They're seen across multiple different systems. So we have clear evidence supporting this correlation. And in the US, the sector that uses the most antimicrobials is the livestock system, right? So 60 to 75% of all medically important antimicrobials are used in livestock. And there's lots of different reasons why managers are using antimicrobials, which we'd be happy to talk about later if anyone's interested. But one of the primary reasons is because they want to protect their herd, right? They want to prevent outbreaks. And they want to particularly prevent outbreaks of very severe infections, so virulent pathogens. So if we have this pattern between virulent pathogens and drug use, and we know that increasing drug use leads to increasing resistance, one way to understand broad scale patterns and variation across the landscape, which is just an example across all of the state of Wisconsin of E. coli, not just in those limited sets that I showed you earlier, we see that there's lots of variation because there's lots of variation in drug use in these dairy farms, which dairy farms have the second highest concentration in Wisconsin. First highest concentration is in California. But suffice to say, we have a lot of dairy cows and a lot of farms and a lot of drug resistance in Wisconsin. So if we want to reduce these patterns, we could start to look at the possibility of managing virulence to reduce drug resistance, right? So this is a different strategy than just continuously developing new antibiotics. That strategy is an arms race. We develop new drugs, bugs develop resistance. And we could just continue that arms race perpetually, except it's very costly, both in terms of time and money to develop new antimicrobials. And big pharma has fled the field because there's just no money in making new antimicrobials. And those same barriers inhibit startups from also gaining any traction in that field. So we've kind of hit a dead end as far as developing new antimicrobials. We really need to come up with a new strategy. And one that's been proposed is managing virulence. It kind of goes through the back door to manage drug resistance. And another big push is to reduce the use of antimicrobials in the first place. This is really big push from consumers. There's also been some push for policy. There's lots of loopholes here that I'm also interested to talk about if anyone's interested later on. But we now have a cornucopia of non-pharmacological options that have been proposed. So including prebiotics, probiotics, and postbiotics. And these are all well and good. There are thousands of these on the market, hundreds coming out every week. For the dairy industry, I did a quick Google search and there's just in the last year, 4,000 options. And if you go over to the poultry industry, it's way beyond that. The challenge is these are product development and they're usually short-term case studies, not thinking about the long-term evolutionary consequences of these different products. But obviously they're going to change selective pressures and we need to start thinking about their evolutionary implications as well. So we can revise our question with managing virulence by asking what probiotic, in this case, I'm going to use probiotic for all biotics, probiotic immune pathogen dynamics are even possible, right? And we can do that by, again, opening this with in-host black box. And we start with theory that was developed for leaky vaccines. So a lot of vaccines are going to clear most pathogens, but not all pathogens. So they're going to leave behind a few strains. And this is very applicable to the livestock industry, where the goal is not complete elimination of all other pathogens, it's to bring them down to a manageable level below a certain threshold, right? And this threshold is set at what they can manage at the herd level, but also from public policy, public health concerns, right? As we go across the food chain system. So the nice thing about this theory is that it's going to help us compartmentalize, make sense of all of these different products. So we can start to group them into three basic flavors. One is anti-infection, because that's what it does, right? It's kind of a barrier, prevents the infection from gaining any traction in the first place. The good thing about this is that it prevents multiple infections, right? So it prevents multiple strains from gaining any traction within the host. And this is important because competition among all of those different strains is known to select for higher virulence. So by preventing infections and multiple infections, anti-infection strategies tend to select for low virulent strains. Conversely, if the infection takes hold and then we want to help reduce the growth of that pathogen, that tends to select for these fast-growing pathogens that are trying to grow fast enough and distribute enough propagules into the environment that they can transmit to new hosts and become established in the population. So this tends to select for high virulent strains. The other large sort of flavor of these different treatments is anti-damage or tolerance. And this just helps reduce pathology, right? So it helps the host just not really care that it's infected. The problem here is that this is just a free-for-all for the pathogen, and so it reduces the cost of virulence and tends to select for higher virulence. So we can use this as a guide to plug into our between-host model and our within-host model interactions between probiotics, the immune system, and pathogen dynamics, right? So I'm not going to go into all of these equations, just to kind of give you a flavor of what types of ordinary differential equations we're using. And again, remember the important part that we're doing here is we're not just focusing on within host or between hosts, but we're connecting the two to look at dynamical cross-scale feedbacks between these processes. Okay, so I'll show you the results from this model. Across the bottom, we have this relative change in probiotics. We're increasing the probiotic level. And on the y-axis on the top panel, we have the relative change in prevalence. On the bottom, it's the relative change in virulence. And then across the top, we're going to look at anti-infection, anti-growth, during this other one, which is anti-growth, but with immunopathology. So these are things like cytokine storms that are still causing inflammation that this probiotic can't take down. And then tolerance, right? And it's all relative because we've scaled it so that we can compare the magnitude of each of these different treatments across these different dynamics. Okay. So what you can see, just first takeaway, is that you get drastically different outcomes depending on which of these treatments you go with. And so it matters if you're selecting a non-pharmacological option to think about whether it's preventing infection, growth, or tolerance, right? So that's just one simple guiding rule that shouldn't be that difficult to connect to experimental studies. The second part you'll notice is that you have really different outcomes in prevalence and virulence. And this highlights that you have to think about whether you're managing for the population or the individual, right? So some of these treatments are going to reduce prevalence, right? So if anti-growth is going to reduce prevalence, but we have a skyrocketing virulence. So this is good for the herd, bad for the individual. And that might be something that farmers are willing to take, right? They're going to lose 4 cows versus the entire herd. That might be preferable. If you're trying to manage for virulence, though, you probably want to go for this anti-infection strategy. So this is going to drastically reduce virulence, but you end up having higher infection overall. The worst case scenario is tolerance. In this case, you do very little to prevalence, but you skyrocket virulence. So if we're thinking about drug resistance and virulence management, we're going to be way over here, right? And this tolerance aspect is really interesting because this is probably what's going on in salmonella and poultry. For example, poultry are loaded with salmonella, They don't really care, doesn't really bother them, but they shed it everywhere in the environment. There's lots of different species. Some are more virulent than others, particularly to humans. And because they're tolerant and it doesn't bother them, it spills over into the food chain, right? Okay, so conclusions from part 2. Variation in drug resistance is widespread. This is the same for virulence. I showed you mostly drug resistance patterns. We know this is true for virulence. And this likely helps us understand different patterns in drug usage across different scales, particularly across different managed livestock systems. We've shown in our models that the evolutionary trajectories depend on resource-dependent feedbacks between microbes and the immune system. These feedbacks make the system very dynamical and sensitive to initial conditions, like pathogen dose. So in a high shedding environment, a herd that has lots of infections and shedding into the environment, it's going to expose individuals to higher pathogen dose. That's going to have implications for the evolutionary trajectory of the system, as does the initial condition of immune functioning, right? And this initial conditioning immune functioning means that the system is very pliable, particularly if we want to modulate immunity, right? So we don't want to do something like improve tolerance and just help the immune system cope. We want to help it prevent infection in the first place, if we're trying to manage for virulence, right? And I think the most important part of this is that the model tells us, one, what outcomes are possible, and it tells us which parameters we need to measure empirically. So that if we want to start to build out these probiotic options or biotic options as viable non-pharmacological treatments, we can start to organize them around within these evolutionary framework. And this seems completely logical, hopefully, but most of our modeling and our experimental systems do not take these factors into account, right? In part because if they're looking at individual dynamics, they're simply looking at very short term frames. Very rarely do we have studies that manipulate a treatment and measure simultaneously immune function, pathogen load, and shedding. And that's what the model says that we need in order to understand where we're going from a prevalence perspective and a virulence perspective. And from the first part of the talk, also see that that's really important for understanding drug resistance, because again, these are all competitive interactions among different strains. So we really, really need to dig into the within host aspects of our experiments and our models. Because again, we know that all individuals are not the same in their infection outcomes. And if we want to understand this, from a niche space perspective, then this can help us understand why we have variation in hotspots, both across a landscape, but also in individuals. And this kind of perspective, I have a lot of promise in thinking about this because one, it goes straight back to ecological theory, but also this has come directly out of conversations I've had with managers. So if you talk to managers, they are not convinced that this statistic of 60 to 75% of antimicrobials being used in livestock is the reason why we see high prevalence of drug resistance, both in livestock sectors, or in humans, or in the environment. What they see with their data is that there's something going on about niche space. And so that creates a lot of crosstalk between these different disciplines, which I find really promising. Okay, now, this is the last part of my talk. It's kind of shitty. I hope you're still awake. So in an interconnected world, we know that what happens on the farm does not stay on the farm. And if you live in Wisconsin, this is a super common scene. And it smells amazing. And this is showing a green, really nice greenery here. But the thing is, it happens a lot of time, like multiple times in the season, right? So sometimes there's nothing here but bare soil. And as an aquatic ecologist by training, you can't help think about what this is doing to the lake systems that you love. because there's nothing there to keep it from running off into this beautiful watershed that is Wisconsin, right? But this is just the state of the world, right? We all like cheese. Wisconsin, we love cheese curds. We all like milkshakes. This is all Wisconsin, right? So all these cows produce a lot of manure, and we have no real viable options for it. This is just, it's gonna happen because we have nothing else to do with it. So how do we deal with this really serious challenge that has ramifications across the livestock sector, public health, and environmental ramifications, right? So we went back to first principles, again, consumer resource ecology, and we ask, how can we reduce the spread of drug resistance? Given that manure is being spread everywhere, we can do what we can at the farm level by, say, managing to prevent infection in the first place, to reduce virulence, reduce drug usage. We're not going to eliminate all the resistant pathogens in the manure. It's going to be there. So we really need a two-pronged approach, probably like a 20-pronged approach, but let's start with two. Given that we've done what we can at the farm level, now we're spreading manure, how can we reduce the spread of drug resistance across the environment? So first, we can think about competitive suppression, right? So we could have diverse microbial communities, greater species coexistence that could suppress drug-resistant pathogens. That's awesome. I think we should definitely do that. Probably is going to need a little more help, right? Competitive suppression on its own is not going to be powerful enough given the onslaught and the magnitude of this problem. So again, from theory, we can think about reducing dispersal. Okay, that's going to be really hard because, again, we've just seen that we're spreading manure everywhere. The next thing we could do would be to reduce colonization rate, right? So once these pathogens are spread everywhere, we could manage the soil environment so that they can never take hold. This is, again, kind of one of those anti-infection strategies. It's instead of the infection taking hold on, say, an internal organ in a host, we're now preventing infection or colonization in the soil microbiome. So to ask if either of these are possible, we turn to a stochastic model using the Gillespie algorithm. And here we have two different strains. I know there's mole strains, but that's basically what we can do right now. So we have two strains, a slow growing strain that's efficient and sensitive to drug resistance, or sensitive to antibiotics. And then we ask whether or not this slow growing efficient strain can ever outcompete a fast growing efficient strain. And you'll notice here that we've taken away those fitness costs, right? So earlier I suggested that we usually assume, and all of my previous models assume, that these traits come with high fitness costs. Now we've taken that away and we said, actually, in fact, drug resistance makes you grow really fast. You're sloppy at using your resources, but you just go for it, right? And generally, this fast-growing, inefficient strain is always going to be able to survive at a lower R star and outcompete this sensitive strain, right? We have theory after theory after theory, experiment after experiment after experiment showing that. Two ways that we can reverse this outcome in counterintuitive ways would be to vary dispersal to new patches or to vary colonization success. So I'm going to go really quickly through these slides since I don't have very much time left, but what I'll show you here quickly is just that we have the resource, drug sensitive, drug resistant, and in this case we have global dispersal but low colonization. At first, the drug sensitive strain is doing pretty well. Each of these lines are different patches, but then eventually the drug resistant strain just takes over, right? So again, we go back to a homogeneous, low species diversity system full of drug resistant pathogens. The only way that we can reverse this outcome so that this drug sensitive strain can take over is when we have local dispersal. So we've limited dispersal a little bit more. It's not global anymore. Let's say we could keep it within a farm or within a watershed. and we've drastically reduced colonization. In this case, the drug sensitive is able to colonize more patches than the drug resistant. The drug resistant is still there, but it's not nearly as prevalent and it's in way fewer patches. Okay, so the model says this is possible. We're going to use these predictions now to test this in a field experiment. Skip that. All right, this work is led by Colette Nicodem, it's a postdoc in the lab, she's awesome. And it's again in Wisconsin at our experimental farms. And it's our farm to fork study. So we're doing this all the way across the entire food chain. So we have an experimental farm where we have some plots that are receiving manure application from dairy cows. And then we're tracking this across different soil health systems that are being managed to improve soil health in a number of different ways, which I'll show you in just a second. And then we're actually tracking this across the food chain by looking at forage crops. And some of these forage crops are cash crops and others are actually going back into the food chain. And primarily they're being used to feed cattle. So we have this entire system where we can look at different ways that we can reduce the spread of drug resistance given manure application. So this is an amazing resource that we have. This Wisconsin Integrated Cropping Systems trial, it's adjacent to the Danes Blairy Farm, which is an experimental dairy that's run by UW. And at the Wisconsin Integrated Cropping Systems, they have a gigantic manure lagoon that receives, it just undergoes standard industry policy, procedures for composting and treating manure. And then we have 9 different cropping systems. Again, these are set up to improve soil health. And these are being implemented across the state and across the country through extension agents that are working very closely with farmers. So we can now ask whether we could harness soil management policies to manipulate microbial community composition in order to reduce drug resistance, the spread of drug resistance, and also pathogen spread across. I'm really excited about this. I was hoping that I would have results. They're at the sequencing center because we just finished our first field season starting earlier this spring, but they're just not back yet. So you'll have to stay tuned for that. It's going to be exciting. And to end, I'll just say that I'm hoping that this project also gives me great promise, because if you start thinking about antimicrobial resistance, you quickly get overwhelmed because, one, it's everywhere. And what are we supposed to do about it? Well, by working across these different systems and working with different managers, hopefully we can have these ecologically and evolutionary guided solutions for these managed systems. And this is a really important place to start. I mean, I am an ecologist by training. I love being in the mountains and in wild, beautiful, remote places. But when I really care about those species, what I always come back to are these managed systems, because they have outsized implications for all of our favorite places and our favorite people. So hopefully with this approach, this question will kind of be redundant, think of the past. All right, thank you very much.
Thanks so much for your talk. That was really awesome. I loved it. And I'm super excited to see more work that you do and that your lab does.
Thank you.
So my question is, when you're talking about the sort of competition between different, microbes living in the same system or something, do you consider their competition with one another to be like equal on both sides, or is one of them having more of an impact on the other one? And if so, I'd just like to hear more about how that works in your model.
Yeah, that's a great question. If we go, so back when we were looking at efficient slow growers versus fast inefficient growers, in that case, the fast inefficient grower, which is the resistant strain in this case, has an outsized effect because it's gobbling up all of the resources. And so in that case, it's it always competitively excludes because it exists on a lower R star, right? It pulls the resources down, but it's still doing fine. And that's just kind of the baseline to start. there are lots of different outcomes that you could have, right? There's even facilitation among certain microbes. So certain microbes will break down antimicrobials, and that allows other microbes to kind of have free range in this lovely environment. We haven't considered that in the model, in part because the reason that we started with efficiency and resource use is because there's some of my favorite research is from Alison Lopatkin's lab. She has done amazing work. I think she's at UConn now, but she's done really amazing work looking at these metabolic adaptations that emerge prior to the drug resistance. And those seem to facilitate both tolerance, so pathogens to go into kind of this metabolically deactivated state so they can fly under the radar and not be sensitive to antibiotics. And then it also allows them to deal with drugs to like be resistant as well. And so the challenge that we have with that is that she's done all the sequencing work and she finds that these metabolic genes are overexpressed in clinically important isolates of E. coli. And the question is, what do you do with all that sequencing data? Like, how do you make sense if you were going to go after metabolic genes? How would you know which ones to target? And so we went back to theory for consumer resource ecology, and the way you get those different outcomes is resource acquisition or conversion efficiency. So we started there because if we could start to try those in, we could connect them to genetic adaptation as well. That's kind of the long term. That was a long-winded answer.
Yeah.
Did I get?
Yes, thank you.
We have a question from the chat. Kelvin has two questions. Would it be naive to argue that reliance predominantly on static rather than cytal drugs should minimize the emergence of resistance?
Sorry, can you read that one more time?
Would it be naive to argue that reliance predominantly on static rather than cytal drugs should minimize the emergence of resistance?
I mean, yeah, in general, if you're trying to not kill, but just slow down, that's gonna usually, that should reduce the rate at which resistance emerges. But what we find is that pathogens always figure out a way to adapt through these either static or cytal. And that's in part because they target a very limited number of mechanisms within the bacteria. And so those are really evolutionarily conserved adaptations, and the push has been to expand out our arsenal so that we're not just always kind of going after those in different flavors, static versus cytal. Great question.
And the second question is, growth rate is a fundamental virulence factor in your models. Have you seen any evidence that resistance mechanisms with larger production burden on the pathogen take longer to spread?
Longer to spread. Well, in our models, we have a virulence transmission trade-off, which is very similar. And so that's gonna, the faster they grow, that's gonna come at the cost of transmission rate. So the faster growing ones have higher virulence, because that trades off with transmission, that's gonna limit transmission rates.
So word on the street says you're trying to apply this study to salmon populations.
Oh, yes.
Is that rumor?
Yeah, can I just show you some, just really quick? So this is a project on Vancouver Island that's led by Chloe Foyo. And this area of Vancouver has the highest concentration of salmon farms in Canada. We know that salmon farms use a lot of antimicrobials because they're growing fish at very high densities and stressful conditions. There are investigations looking at antimicrobial spillover into marine fish, but we're interested in understanding if we can track antimicrobial resistance in natural populations. So we have this gradient of 48 lakes that vary in connectivity and distance to these salmon farms. And we're, with this great team of researchers, we are investigating spillover into natural food webs, focusing on three-spine stickleback, which are very well-studied species from evolutionary biology. So we know a lot about their feeding behavior and a lot about their parasites. So this NSF project is primarily focused on understanding helminth parasites. By connecting this to antimicrobial resistance, by doing gill swabs, we'll also be able now to look at potential correlations among parasite infections and antimicrobial resistance, and look at how that varies across the males and females infection. And then one really important aspect of these fish is that they have a very important immune response that's really obvious when we dissect them. So it's fibrotic. So if you open them up and they have parasites, their entire system is just this web of fibrotic tissue. So preliminary data show that there's a strong correlation between distance to fisheries and the prevalence of antimicrobial resistance. But we have a couple of outliers in this system. So one of these populations, so this group, these patterns could be driven by competition among microbes, and our preliminary data suggests that there's a pattern of high antimicrobial resistance in populations that have low microbial diversity, and the opposite with high microbial diversity. And then this pattern, which is also a group of outlier lakes, could be driven by resource competition that again changes these dynamics between the gut microbiome or the other microbiome that's hopefully competitively suppressing, and this release of competitive suppression with more resources. So that's an exciting project that I really can't wait for those results as well. Thanks for asking. Yeah, thank you.
They're great talk, Jessica, thank you. Where you have your, like the anti-growth, anti-infection, I guess, modes of the probiotic use, is there a place in sort of farm management, whether it's poultry or cattle, where you could use one that drives one mode, right, and then follow that up sequentially with another, so like maybe you're dropping the virulence but increasing the prevalence, and then you're doing the inverse?
Yeah, that's a great idea. I don't know that any studies have done that actually, because again, that was all developed for vaccines, and usually you're just trying a vaccine, right? So that's a really great point. And that could be done really easily if you have the pre, pro, post biotic.
Right.
Oh, yeah, I really like that. Should do that in your system.
Nicole has her hand raised in the Zoom. You can go ahead and unmute.
Hi, I'm at the vet school, and I'll preface this with it's a loaded question, and I apologize if there are veterinarians in the room. My research experience previously involved diet types and pathogen infection in dogs and cats, and now deals with diet processing for dogs. And I'm curious what your thoughts are about the role of raw diets in dogs and cats and pathogens and resistance and cross-species infection. You know, we're always kind of taught raw diets are the boogeyman, and all pets, you know, that eat raw diets are going to get pathogenic infections, and it's going to spread to humans and everyone's going to die. But, you know, we don't actually see that in clinical practice. I would say it's pretty rare. I make the argument that I would say that raw diets are no longer a fad, and the fact that we see pathogen contamination in both ultra processed diets and minimally processed diets, and you know, the data shows that these infections are pretty scarce. We often suspect that a lot of times dogs or cats are present just with diarrhea or GI upset, that you know, it's related to the food, and that infection is never confirmed, and the pets are given antibiotics and sent on their way. What are your thoughts here about how those differently processed diets potentially play a role?
First, that's an awesome question. I'm glad you're thinking about that. So for raw diets specifically, I will say that the vet school and research in livestock and these managed populations, they have amazing data because they care about questions like this, right? And because they have the resources to be able to connect all the way across. And I think they're really underutilized. There needs to be like more cross-disciplinary research because everybody has different tools to be able to answer these kind of nuanced questions. It's awesome that you all are so closely related to the vet school or geographically located. So I don't know about raw diets specifically. I can say that the thing it's always going to come back to exposure levels and immune functioning. And so if there are the way to get at that mechanistically would be for studies to look at raw diets versus processed diets and how that affects immune functioning. And then if you're going to do that, you need to track your favorite flavor of immune functioning. So cytokines are always a great place to start. But within your system, there's going to be a panel of different immune metrics that are going to be the best. And if you can track the functional response of how immune function varies across a gradient of these two different kinds of resources, so not just yes or no, but across a gradient, that's how you can start to connect the mechanistic interactions, both just statistically and in a model. And then if you can do the same thing, looking at pathogen load, but also transmission into the environment, those would be the metrics that you'd want to go after. Yeah, thanks for your question.
Thank you.
How do you consider like microbiome as a factor, for example, when you're looking at drug resistant individuals that are like optimal for like within a host versus like system wide? Do you look at microbiomes as a factor, like competition with other bacterial things in the,
Yeah, when you mean system-wide, do you mean,
like when you were talking about host specific infections, and then like it was in the first part
Okay.
versus like mutations or mutants that are better suited for like transmission between hosts?
yeah, absolutely. So, I mean, I'm talking about two species here because it's what most of my models can handle, but these data are definitely taking into account the microbiome, the overall diversity of the microbiome, because the idea here is that we don't just have a sensitive strain and a drug resistant strain, but the microbiome as a whole is going to differ, and that's what's going to competitively exclude these virulent or drug resistant strains. So absolutely, I just haven't shown you those data, in part because they're still in the sequencing center. But yeah, those are definitely patterns that we're going after. And again, back to livestock research, excellent research on this in poultry with salmonella. So the best evidence we have for competitive exclusion, all of these different variable, like different mechanisms of competition or facilitation, those are done in livestock systems. Yeah.
So that's all the time we have. One more round of applause.
Thank you, but I need to do that. Can I just check this?